In our previous classes, we already covered Mendel's monohybrid and some of the laws, and we just plotted Punnett square and just crossed a single type of trait in your monohybrid cross. Same as in dihybrid cross, we also take the traits, but the difference is here we take two type of traits because the topic is your dihybrid cross. So di means two. So we take two character over here. So according to Mendel, he take two type of character of a plant. The plant is your pea plant, and the characters are, one is your round and yellow seed, and the another plant, it is wrinkled green. So he just took two type of plants, and it is the phenotypic appearance. That means physically one plant is your round and yellow, that means the seed is your round and the color of the seed is yellow, and the next plant's seed is wrinkled and the color of the seed is green. We just cross two traits, that means it is your one trait, it is your two trait. We just cross two type of traits over here. So the genotypic ratio is your capital R, and for the yellow, capital Y is crossed with small r with small y. So he just crossed it. So it is your parental generation. So this is your parental generation. Nextly, the crossing means firstly the gametes formation takes place, nextly the crossing. So the gametes. So gametes are, one is your capital R capital Y with capital R capital Y. There is no other possibilities. This is a single. Nextly, this is your small r small y is with small r small y. Nextly, it is your gametes. Gametes are formed. Nextly, the crossing which takes place means the resultant is your first filial generation. That means the F1 generation is going to form. So for the F1 generation, we just cross the terms first. We just cross the gametes over here. We just plot a small Punnett square because only two traits are there, and wrote like that. Your capital R, capital Y, capital R, capital Y is crossed with small r, small y, and small r, small y. Next is the crossing takes place. So whenever you cross, remember one thing, that the capital letter is wrote first. The reason behind it, the capital letter is the dominant character and the small letter is the recessive character. So dominant character is look first. That means whenever you check a trait, the dominant character is first and the recessive character is suppressed down. So the expressive character is wrote first. So you just wrote the capital letter first. So we just cross small r with the capital R, and here is the resultant, capital R, capital, small r, cross with your capital Y, small y. Nextly, the capital R, small r is crossed with capital Y, small y. Same as here, capital R, small r with capital Y, small y, capital R, small r with capital Y, small y. So this is the resultant of your F1 generation. Nextly, the selfing characters. In your previous classes, I told you that in important terms, selfing means the selfing between the two resultant of a single generation is takes place. So your single generation means your first real generation, and the crossing means the resultant over here, and we just cross two resultants again. We just cross two resultant again. So we just took randomly two resultant and again cross it. So selfing is takes place. In selfing, we just cross it. Capital R, small r is crossed with capital Y, small y. Here also capital R, small r with capital Y, small y. There is a trick. You just cross like that. Capital R is crossed with capital Y first. We just wrote the gametes over here again. 4 gametes are formed. So capital R is crossed with capital Y. Again, capital R is crossed with small y. So capital R is crossed with small y. Nextly, the small r, here is a small r, is crossed with your capital Y. So it is small r is crossed with your capital Y. Nextly, the small r again crossed with your small y. So here also the small r is crossed with your small y. As same as here also the capital R is again crossed with your capital Y. Capital R is crossed with capital Y. Again, capital R is crossed with small y. Capital R is crossed with small y. Again, small r is crossed with capital Y. And small r is crossed with your small y. So here is the resultant of the selfing. Nextly, we took the resultant, that means your gametes, and nextly plot a Punnett square again. And this is your F2 generation. We already plotted a Punnett square for the F1 generation. Nextly, the selfing takes place. Nextly, for this, for the gametes, I took over a second Punnett square, which is your F2 generation. So here we plot again a Punnett square, and it is your Punnett square for your F2 generation. So this is your second one, and here it is your third one. Nextly, we just plot the resultant over here. That means the gametes. The gametes are, you may wrote it just here for your male plant or for your female plant over here. It is your capital R, capital Y. Nextly the capital R, small y. Nextly the small r, capital Y with the small r, small y. Here also the same. In this side also, capital R, capital Y with your capital R, small y, small r, capital Y with your small r, small y. Next, the important thing, how we fill the box. And the only one trick which is look quite easy for me is that you just cross a single term to all the terms and fill serially one by one. It is only one easy trick. You just cross it. You just look over here or take a pen and paper and just cross. How I just cross over one by one. You just cross one by one. You just cross capital R, capital Y with your capital R, capital Y first. So the crossing is takes place one over here. Capital R, capital Y with the capital R, capital Y. Nextly, the capital R, capital Y with the capital R, small y. Here is your capital R with your capital R. Nextly, the capital Y with the small y. Whenever you wrote, first wrote the capital letters. Nextly, the capital R is crossed with capital R with your small r, next the capital Y with your capital Y. As same as the last one, capital R, small r, capital Y, small y. Nextly, the capital R is crossed with capital Y, small y. Nextly, the capital R is with small y. Capital R, small r is crossed with capital Y, small y. Capital R, small r is crossed with your small y. Nextly, the capital R, small r is crossed with capital Y. Nextly, the capital R, small r is crossed with your capital Y, small y. Same as small r is crossed with your capital Y, small y. We just cross over here. I just cross small r, capital Y with small r, capital Y, and the resultant is here. Nextly, it is your capital Y. Nextly, the smaller is crossed over here, and the resultant is capital Y, small y. Nextly, the capital R, small r is crossed with capital Y, small y. Capital R, small r is crossed with small y, small y. Small r is crossed with capital Y, small y. Nextly, your small r is crossed with small y, small y. So we just cross every terms. Nextly, it is according to Mendel, it is the genotypic terms. We just wrote the phenotypic terms again. So for that, you just look a thing over here. The capital R is represented the round cell. The capital Y is represented the yellow. That means the color. Next, the small r is represented for the winged, and small y is represented for the green. So according to this, you just plot the names of them. So capital R means your round cell. Capital Y means yellow. Nextly, round. Capital Y, yellow. Nextly, it is also your round, yellow. Here also round, yellow, round, yellow, round, yellow, round, yellow. Simultaneously, you will also note down it. So here also it is your round, but small y is present, so it is your green. So it is your small round green. Next it is your round yellow. It is also your round, but it is green. Next it is round yellow. It is also your smaller means your winged, and Y means your yellow. Here also your winged, yellow. Here also your round, Y is present, so it is your green. Nextly, it is your wrinkled. Capital Y is present, so it is yellow. Nextly, small y means your wrinkled, and small y means your green. So we just fill the phenotypic names of that plant. So here one thing left. What is your phenotypic ratio over here and what is the genotypic ratio over here? So we just plot the F2 generation over here. So this is your F2 generation. So after the F2 generation, we have to write the phenotypic ratio and the genotypic ratio. So it is also quite easy. You just look the Punnett square clearly first. In Punnett square, you found 4 type of traits. That means 4 type of phenotypic means one is your round yellow, another one is your round green, next one is your wrinkled yellow, and last one is your wrinkled green. So first one is your in phenotypic ratio. First one is your round yellow. Next one is your round green. Next one is your wrinkled yellow. And wrinkled green. So we just get these 4 type of physical appearance. So we just take it as ratio. So phenotypic ratio of round yellow. Just count the round yellow first. So in the Punnett square, here is one round yellow, number 1. It is your 2 round yellow. It is your number 3 round yellow. It is your 4 round yellow. It is your 5. It is your 6. It is your 7. It is your 8. It is your 9. So you found over here 9 traits are round and yellow. So here we simply wrote 9. Nextly, the round green. We just count it again. Round green. It is one round green. It is your number 1 round green. Nextly, number 2. It is also round green. Nextly, it is your 3. It is also the round green. So we found 3 round greens over here. Nextly, the wrinkled yellow. Let's count it. The wrinkled yellow. Here is one. Wrinkled yellow. Nextly, here is also one. Your 2 number wrinkled yellow, and here also another one. It is also your wrinkled yellow. So you also found 3 wrinkled yellow traits. Nextly, the wrinkled green trait. It is only one wrinkled green trait. So it is the wrinkled green trait. It is the only one. So here I wrote over there wrinkled green. So the phenotypic ratio of diabetic cross is your 9:3:3:1. So it is we found by plotting the F2 generation. So it is your phenotypic ratio. Nextly, the genotypic ratio. And before going to the genotypic ratio, I also clear one thing that if you look carefully the Punnett square, we found that if we plot a simply triangle over here, the path taken by the triangle, that means you just all the traits where the line is crossing, everyone is your rounded yellow, and another one it is a small triangle over here. Everyone is your wrinkled yellow, and the rest 2 are is your wrinkled green and another traits, that means your round greens. So this is the gist of your phenotypic ratio and the resultant of your 9:3:3:1, and which is always known as a phenotypic ratio. Nextly, the genotypic ratio. I already wrote over here the genotypic ratio. So if we look after the genotypic ratio, it is somehow like that. That means your 1:2:2:1:2, 1:2:4:2:2:1, 2:1. That means it is a long term. For this, you just look again the plotting Punnett square. Here you found the trait capital R capital Y with capital R capital R with capital Y, a single one, which is this. This is your trait of your capital R with capital Y, capital Y. I wrote over here. Nextly, two traits of capital R small r with capital Y. Here is one, and again in this line you just found another one. Here is the capital Y. In this way, you just found all the traits. I already wrote the traits. You just found it. It is your homework. Nextly, you just look all these things. This line, you just count it. That means 1, 2, 3. You just count it. The resultant is your 9. That means 1, 2, 3, 3 plus 2, 5, 5 plus 4, 9. So 9 is the trait of your round and yellow. So you just found over here 1 is to 2 is to 2 is to 4. It is the first. Nextly, the 3. That means the first 2. That means your this one is your 3. Nextly the 3. Nextly the 1. That means your 9 is to 3 is to 3 is to 1. The phenotypic ratio, and this is your genotypic ratio. So according to this, it is your 1 is to 2. Nextly 1 is to 2. Nextly the 1. So it is the genotypic ratio, but we just sequentially wrote it as, that means your genotypic ratio is your 1 is to 2 is to 1 is to 2 is to 4. We just wrote this side first. Is to 2 is to 1 is to 2 is to 1. So this is your genotypic ratio. In exam, mostly genotypic ratio is not much required, but it is just a clumsy thing, but easy thing. And the phenotypic ratio is the most common question, and the diabetic cross is also a most important term and the factor, which is you have to understand it clearly. Hope this concept is helpful to you. Hope this session is helpful to you. If the session is helpful to you, just hit the like button and wrote your comments over here how you feel after the session. So till then, stay tuned. Thank you.